I want to talk about our efforts to link biochemical assays and cellular efficacy data, or to cellular efficacy, and related to inhibitors of the coronavirus main protease. This is based on my student's PhD defense, and a lot of the work was hers, or most of the work was hers, and Lulu Kang, a mathematics professor, was a major collaborator. Okay, so you're all aware of the different coronaviruses. One notable thing I want to mention is the Middle Eastern coronavirus being having a very high mortality rate compared to the other coronaviruses. And you're all aware of the goal of targeting the coronaviruses by inhibition of the main protease, which plays a major role in the viral life cycle. I won't go into too many details to make sure that there's enough time. As part of ASAP, what my group was doing is trying to help connect the results from in vitro biochemistry assays to make predictions about what would happen in cellular assays. The cellular assays are much more expensive and require much higher biosafety levels. So it was advantageous, or it was considered advantageous to be able to make predictions of cellular data assay results prior to doing so, and by using biochemical assays instead. In a typical biochemical assay, is we increase the concentration of a compound, we'll get a concentration response curve, which is monophasic. That is that the activity of an enzyme, when you add more of an enzyme inhibitor, will reduce as you increase that in a sigmoidal curve that can be fit with this equation called the Hill equation. And that's the typical way that these data are analyzed. However, it's been observed that with the MERS coronavirus, that these type of behavior is not always observed. What happens is, as small amounts of compounds are added, there's actually an activation phase where the activity of the main protease will increase prior to decreasing with the addition of additional inhibitor. This behavior was recapitulated with the SARS-CoV-2 main protease when a mutant was created that weakens the dimerization interface between the monomers of the enzyme. Then the question that we had, which inspired us to look into this project, or to do this project, was to understand how we can interpret the data from this assay to make some prediction of what's happening in a cellular assay. There are a number of possible procedures that could be attempted to analyze the data from such a biphasic curve. Two that we considered and were implemented in ASAP prior to our being involved, or at least one of these was in the control IC50 was implemented prior to our involvement, is basically we can split this curve into an inhibition phase and or activation phase and an inhibition phase. And if you consider the inhibition phase only, that would be, and then look at the IC50 of that we call that the inhibition PIC50. That's on the left-hand side. And then another possibility is to just control or use the Rmax value, or the maximum of the curve, being based on the negative control. And that's what we refer to as the control PIC50, and that's on the right. You can see that there's a shift to the right of the control IC50 relative to the inhibition PIC50 for the analysis of these biochemical concentration response curves. It is not clear, though, even if we can do this fit, whether this is meaningfully connected, or was not clear at this time, whether it's meaningfully connected to the cellular activity of the inhibitor. So the hypothesis that we had is the cellular activity of Mpro inhibitors is driven by the potency of the against the dimer, not of the monomer. And that was how we thought we could explain the data that's been observed in previous studies, but there was no model that was fit to that previous data. And the model that we developed is an enzyme kinetics model that involves not only standard binding to an enzyme, but also the dimerization of the enzyme, and also the binding of substrate and of inhibitor, and there being 9 total states of the enzyme and enzyme substrate or inhibitor complex. And there's transitions between these different states. I'll talk a little bit more detail about some of these parts of the model. Well, oh yeah, okay, I'll just run through some of it. Like you can have a monomer of the enzyme that binds to another monomer to get a dimer. And then you can have a binding of a substrate to get the dimer substrate complex. A second substrate get a dimer substrate complex. And if you have these complexes, you can either dissociate the substrate or you can have catalysis to lead to 2 product here. Every enzyme substrate complex has the ability to form catalysis or a catalytic rate associated with it. And there's also affinities associated with the inhibitor. That's what these symbols are, that with the X are describing inhibitors. And you can also have mixed complexes where you have a substrate in one of the subunits and an inhibitor in the other subunit. So we made a rapid equilibrium model. So that's making the assumption that all of these species are in a rapid equilibrium, and this equilibration occurs at a much faster time scale than the catalysis. This model is associated with a number of parameters. Actually, a lot of parameters. There are thermodynamic parameters for the binding of the substrates and of the inhibitors and of the monomer to itself to form the dimer. There are also some constraints inherent in this schematic because of these closed loops. You can see that mathematically, if you try to multiply certain quotients together, or some of these equilibrium constants together, they should satisfy these constraints. Other information that will allow us to determine concentrations of individual species are conservation of mass constraints, that you have a certain amount of monomer that you put into solution, initial amount of monomer, and then each of these species will have a certain amount of monomer, and similarly with substrate and inhibitor. And the velocity of the catalysis is equal to a catalytic rate times the concentration of each of these complexed species. So what we were hypothesizing is that in a biochemical assay condition, you have a relatively low amount of protein, and you have a lot of monomer sitting around. And that's why when you add some of the inhibitor, you cause some dimer to form, and then you cause some catalysis to happen. And that this dimeric form is essential for catalysis, or this is believed to be essential for catalysis in other publications. And we hypothesized is that when you have a cellular condition, the cell is going to produce a lot of the protease as instructed by the virus, and the monomer is not going to be as relevant to its or to consider. And we'll just consider what happens if we only have a dimer around, basically cut out the left side of the model, and then make a prediction of what the curve would look like if you only consider the dimer states, and then vary the amount of inhibitor and keep all these constants the same. So in order to evaluate this hypothesis, first we had to develop a way to fit to data and to estimate parameters. Then we had to apply this to multiple inhibitors. Then we looked at the estimated PIC50 from the model and then the cellular P EC50 of the series of inhibitors. And this will all be discussed quickly. So in order to construct this model, well, first of all, I want to talk a little bit about why we used a Bayesian approach, and then what we did to fit this model. So first of all, we saw that you can fit this model to lots of different data. So this is a concern that's always present when you have a complex model, is that you can fit it to lots not a lot of data, is that you can overfit the data that you have and not have a clear understanding of the parameters. So what we did was we tried to fit to a lot of different data, and this is data from a paper published in 22 by Nash et al. And we saw that we could indeed find a way to fit this model to the data, but we weren't clear about whether these parameters were determined, how well these parameters were determined. And this motivated us to move beyond a standard nonlinear regression approach to consider a Bayesian regression approach, which we also had been a major effort in my research group. So the Bayesian regression, briefly, is based on Bayes' theorem, and it talks about the probability of parameters given a set of data. That's probability of parameters given these observations being proportional to the probability of the prior probability of the parameter times the likelihood of observing the data given the parameters. The advantage of this approach is that you can incorporate knowledge about the parameters, for example, from other experiments, into the prior. And also, this gives you an idea of rather than just saying you have one set of parameters that fits the data, you have a range of parameter values, and they could be correlated with each other, and they could be multimodal, but there are multiple ways, or this could account for many ways that parameters can fit to specific data. And when you see the range of this posterior probability, that is a way to quantify the uncertainty of your parameter, or how well you know what that parameter value is. So the data we analyzed first was from this publication from the Lewis group. And they had postulated the existence of this mechanism, but not actually fit this model to the data. I also want to mention that this model is not, we don't have an analytical solution to this model. Like, we can't find exactly what the values of the concentrations are based on a direct formula, but rather we used a computer numerical solver to find the solution, the numerical solution to the equation for this model. So we have all the parameters that we deal with are all the parameters I mentioned. There's also an experimental noise parameter, and we said that we fit to data of MPRO mutant and wild type at the same time, but we said some of these parameters are shared between the enzyme variants and some of them are distinct to the mutant or the wild type. And then we fit this model to data, and we saw whether it could fit all the data sets. It did fit all the data, just like when we did the nonlinear regression. The useful aspect, or the particularly useful aspect, is that you can get an idea of how well each of these parameters are known. And some of these are quite tight parameters, and we can also know that these are unimodal or largely unimodal. You can also tell whether they're skewed. This describes our extent of knowledge of each of these parameters. And based on fitting to the paper from the Nash et al. Paper, we were able to find posterior probabilities for many of these parameters that are fairly tight, except the affinity of the inhibitor to the monomer is not clear whether it's a high affinity or a lower affinity binding, because it's just not clear from that data that we were provided. There's other parameters which are just, we have no idea what they are, and these include the rate parameters actually. And we can also look at the correlation between parameters, and this means that if these are highly correlated, that means that when this parameter is low, then this other parameter is also low, and vice versa. There's a negative correlations. It's meaning that the data can be fit by sets of parameters where this is high and this is ki is high and log k SDI is low, for example. But many of these parameters are fairly independently known from each other. So that was our first step, is to apply this regression method to get an idea of what these parameters are. And then we wanted to see whether we could work with multiple inhibitors. That was the next step that we considered. And we started analyzing data that was collected from Chaim Bar's group at ASAP. And some of these data were only with the enzyme and the substrate at different concentrations, and some were with different concentrations of inhibitor. Or sorry, of these, all these series have different enzyme and substrate concentrations, and then the concentration of inhibitor is given by the position on the x-axis. And so we first just fit the top of the model that doesn't include any inhibitor complexes to the enzyme substrate, or to the enzyme substrate data. And then we fit, or we consider the data that also included inhibitor or different concentrations, the full model to concentrations with inhibitor as well. And when we consider multiple inhibitors, we would have unique values of parameters that relate to the binding of each inhibitor and of the interaction between the substrate and inhibitor. These would be distinct, whereas these parameters would be shared across all the different data. And with this approach, we are able to fit to a reasonable set of inhibitors of where there are different positions of, or different positions of the key features of these curves. So this gave us confidence to apply this more broadly across different systems, and that the model was suitable for interpreting a lot of data from this system. Here's an idea, or this gives you an idea of what can be learned when you fit to multiple data sets at once. If you only consider one enzyme substrate data set and with one particular inhibitor, these are the posterior densities, and on the right is if we fit to 13 different, or data from 13 different inhibitors. You can see that all of these densities are narrower, and so that gives you a clear idea what all these values of these parameters must be in order to fit all these data. So that after being able to fit the multiple inhibitors, data from multiple inhibitors, then we wanted to see whether our hypothesis was correct, that we would be able to look at the correlation between the predicted PIC50 and the cellular PEC50s. And just as a reminder, we only looked at the dimer in the model. We would make a curve or simulate a curve based on that, fit or determine the PIC50 of that curve, and then we looked to see how it would correlate with the cellular PEC50s. And yeah, here's another way to visualize that. And I'm providing some details I won't talk about, or some details here I won't spend a lot of time talking about. But in summary, if we look at we can look at multiple ways to calculate IC50s, and we can compare them to each other. So we can look at, as I mentioned, the inhibition phase, the PIC50, the control PIC50, where we set the maximum value of the response to the negative control. And then there's the dimer-only PIC50. And these are all contrasted, or these are all plotted against the cellular PEC50s. We also plot them against each other, so you can get an idea of how like the dimer only compares to the control. They're very highly correlated. All these biochemical assays are quite highly correlated, although there is a bit of a more of a discrepancy between the dimer only versus the other two than the control versus the inhibition. Here's the control versus inhibition, dimer only versus inhibition, and dimer only versus control. It's a little weaker correlation than if you compare the control versus inhibition PIC50. The key point here is that if you look at the correlation metrics, you get the best correlation metric with the cellular PEC50s. If you look at the dimer only, although they're comparable if you compare the inhibition PIC50 and the dimer only PIC50. And if you look at other metrics, the Spearman, Rowe, and Kendall-Tau are more about rank correlations. You get better ranking with the dimer-only PIC50 than the inhibition PIC50. But it's also noteworthy that the inhibition PIC50 is not bad. It's not a bad place to start if you're trying to predict what the cellular activity of a series of compounds that permeates the cell is. We also looked at error metrics, and the error metrics show that the dimer-only PIC50 has a comparable quality to the inhibition PIC50 if you talk about an adjusted root mean square deviation that that makes the means equal to each other. We also had a hypothesis that maybe the 50% inhibition is not sufficient to have a cellular effect, and we may have a better effect if we look at the 90% inhibition, and we want to compare whether the dimer only or the biochemical IC90s were better correlated with the PEC90s than the 50 values. And in this case, we see that there's a better correlation between PIC90 and cellular PIC90 from the Pearson R, as well as these rank correlation metrics, and in terms of error as well. Although, again, we see that the effect is statistically significant, but not so dramatic that it's not useful to use the other data fitting procedures. So in conclusion, or in summary, we were able to construct this kinetic model that was able to fit the data from both SARS-CoV-2 and MERS-CoV-2 MPO, and that we were able to estimate dimer-only PIC50s and IC90s, and they're highly correlated with the cellular PEC50s and PIC90s, and that it's better, statistically significant improvement, but maybe not so dramatic that the other approaches are useless. Thank you for your attention.